Good day, Honors Biology students. Welcome to video lecture episode 21. Now, if you remember correctly, we had started getting into semi-conservative replication, in other words, the means in which DNA replicates. Well, let's go ahead and let's talk about the main stages of semi-conservative replication. Semi-conservative replication has three main stages, the first being unwinding, which is then followed by base pairing, followed up by joining, okay? Joining together, that's what I mean. If you look at the image on slide 10, you can see what I'm talking about there, where you have the parent strand up there, then it sends off two of the templates which connect with these other strands that they make through the process of semi-conservative replication. And then ultimately we end up with 4 new strands at the bottom as it replicates. Alright, so let's talk a little bit about unwinding. To start this stage of unwinding, the enzyme DNA helicase unwinds and unzips the DNA strand. In some ways, it's similar to unzipping a jacket. DNA helicase then breaks the hydrogen bonds as it unzips the double helix, which leaves 2 single strands of DNA. While the 2 strands are separated, proteins called single-stranded binding proteins associate with the DNA and keep the strands separate during replication. You can see this represented on slide 12. You can see the helicase as well as the SSB proteins, that being the single-strand binding proteins. All right. Now let's talk about base pairing. Next up we have base pairing. Another enzyme comes to help at this stage, which is DNA polymerase. DNA polymerase catalyzes the addition of the appropriate nucleotides to the new DNA strand being formed. In DNA, there are 4 bases: adenine, guanine, thymine, and cytosine. These bases are written with 1-letter abbreviation. For adenine, A; for guanine, G; for thymine, T; and for cytosine, C. All right. Those 4 bases are paired up, with A being paired to T and G being paired to C. This is always the case. A never pairs with C or G, just as C and G never pair with T. Just something to keep in mind. All right. Now, DNA polymerase begins adding the nucleotides at the 3' end. As we further discuss this stage of DNA replication, look back at the figure that I showed you on the previous slides with images, just to help you out, okay? Just so you can keep an idea of what's going on in this process. Now, if you look at slide 17, you'll see a particular image here that I want you to look at in connection here, okay? Notice that each of the 2 template strands is formed slightly different from one another. The name of the strand on top is called the leading strand, with the one on the bottom being called the lagging strand. So you can see that on slide 17 on the image there. Now, the reason they call them leading and lagging, you'll see a little bit visually on that image, but we'll talk about that a little bit later as well. Now, the leading strand is continuously built by the addition of nucleotides to the 3' end. The lagging strand, on the other hand, which is elongated from the replication fork, is synthesized discontinuously. In other words, discontinuously doesn't mean it's not consistent. It goes in like portions, stops, gets filled in, goes in portions. Now, it's synthesized discontinually into small segments, which are called Okazaki fragments. Okay, I know that's a little hard to see on the image, but it is included there. The fragments are then later connected by the enzyme DNA ligase. So if you think about that right now, we've already talked about at least 3 different enzymes that are involved with semi-conservative replication as DNA is replicated. Now, each of the Okazaki fragments are about 100 to 200 nucleotides long. With one strand continuous and the other discontinuous, this process is described as semi-discontinuous as well as semi-conservative. All right. Now let's talk about the last stage, and that's joining. The leading strand, remember, is synthesized continuously, but keep in mind in eukaryotic DNA replication, there are often many areas along the chromosome where replication begins. When DNA polymerase comes to an RNA primer, it will remove the primer and in its place DNA nucleotides. All right. Now, once the primer is removed and the nucleotides filled in, DNA ligase links the 2 sections together, thus completing DNA replication. All right. So that we've basically talked about that process of semi-conservative replication fairly quickly, but I think that gives at least a good summary understanding of it. If you have any questions, you can leave them in the comments, send me emails, and I'll be more than happy to talk about this on a one-on-one basis if you need to. All right, well, now let's compare DNA replication in eukaryotes and prokaryotes. Eukaryotic DNA unwinds in multiple areas as the DNA is replicated. Each individual area of chromosome replicates as a section. The length of the section can vary from 10,000 to 1 million base pairs. All right. The result is multiple areas of replication occurring simultaneously, which look like bubbles, as you can see in the image on slide 23. Now let's just keep looking at that image because I also have it on slide 24. You see how prokaryotic DNA is circular. Do you see that? If you take a look on the image on both slide 23 and slide 24, Prokaryotic DNA is circular, and just as eukaryotic DNA goes in two directions, okay, so similar in going the same direction, but you can see it's a bit simplistic. It is shorter, as you may deduce from the image, and the DNA remains in the cytoplasm, not packaged in the nucleus, and we did mention this already before. All right, ladies and gentlemen, this go ahead and concludes our discussion for Chapter 12, Section 2. If you have any questions, you can leave them in the comments below. You can send me email. Have a nice day, and I do hope this lecture has been helpful. Take care. Have a nice day. Bye.